Dear students, in this session we are going to discuss sulfur cycle and phosphorus cycle. Sulfur cycle. It is the circulation of sulfur in various forms in nature. Sulfur is present in all living matter as a component of amino acid cysteine. The sulfur reservoir is mainly in soil and sediments, either in organic form or in inorganic form. Organic sulfur is present in coal, oil and peat, where inorganic sulfur deposits are present in the form of sulfate, sulfides, organic sulfur, etc. in pyrite rock and sulfur rocks. And the sulfur from these reservoirs are released by weathering of rocks or by erosional runoff or by the decomposition of organic matter. And as a result, sulfur will be carried to terrestrial and aquatic ecosystems. The sulfur cycle is mostly sedimentary except two compounds which are gaseous, that is hydrogen sulfide and sulfur dioxide. In the biosphere, sulfur is present in three forms: either elemental sulfur or inorganic sulfur or organic sulfur. Examples for inorganic sulfur is sulfate in aerobic soil and sulfide in anaerobic soil. And examples for organic sulfur is amino acid in plants and animal residues. So this is sulfur cycle in brief. In brief, sulfur-containing proteins are degraded into their constituent amino acids by the action of soil microorganisms. From these amino acids, the sulfur is converted to hydrogen sulfide by another series of microorganisms. The H2S formed will be converted to sulfur in the presence of oxygen and then to sulfate by a group of bacteria known as sulfur bacteria. Eventually, the sulfate again becomes hydrogen sulfide, which may either get rapidly oxidized to gases that may dissolve in water to form sulfurous and sulfuric acids. This may contribute to the formation of acid rain that can harm sensitive aquatic organisms and marble monuments. So there are different processes involved in sulfur cycle: oxidation of sulfur, dissimilatory reduction, sulfur assimilation, desulfurization, sulfur reduction, sulfur disproportionation, etc. So what is sulfur disproportionation? This is also known as dismutation or inorganic fermentation. This is an ecologically and technologically important part of sulfur cycle, where a sulfur compound, a single sulfur compound, serves as both electron acceptor and electron donor. Assimilatory sulfate reduction. This is another process involved in sulfur cycle. Algae and many other heterotrophic microorganisms, they can assimilate sulfur in the form of sulfate. Since they cannot directly uptake sulfide, since H2S is highly toxic, during their sulfate assimilation, the reduced sulfur is immediately reacted with an acceptor, generally serine, the amino acid serine, which upon reaction with the reduced sulfur will yield cysteine, the amino acid which contains sulfur. Cysteine is thus formed. The next important process in sulfur cycle is organosulfur decomposition or desulfuration. Desulfuration of organic compounds generally occur in soils and sediments whereby mercaptans and hydrogen sulfide are formed. So what happens during desulfuration or organosulfur decomposition? The first step is the degradation of proteins through proteolysis by microorganisms, and during this process amino acids containing sulfur will be liberated. Heterotrophic bacteria can use sulfur containing amino acids, and upon their metabolism hydrogen sulfide will be released. For example, cysteine desulfurase enzyme will convert cysteine to pyruvic acid, and along with this hydrogen sulfide and ammonia will be formed. Some bacteria such as desulfotomaculum can reduce sulfates to hydrogen sulfide. In marine environments, the major decomposition product of organosulfur is DMS or dimethyl sulfide. It is formed from DMSP or dimethyl sulfoneopropionate, which is used and utilized by bacterioplankton or floating bacteria as a sulfur source. So, in marine environments and in sediments and soils, hydrogen sulfide, dimethyl sulfide, mercaptans etc. are formed, which escape to atmosphere, where upon photooxidation they will become sulfate. The various reactions involved in sulfur cycle can be either oxidative sulfur transformations or reductive sulfur transformations. Coming to oxidative sulfur transformations, hydrogen sulfide may get subjected to microbial oxidation either under aerobic conditions, or it may be phototrophically oxidized under anaerobic conditions. Chemolithotrophic microorganisms such as Beggiatova, Thioploca, Thiothrix, etc. can oxidize hydrogen sulfide to sulfur, and the formed sulfur will be deposited in cells as sulfur globules. In the absence of hydrogen sulfide, these organisms use these sulfur globules and they oxidize sulfur slowly to sulfate. Some species of thiobacillus, which are not highly acid tolerant, they oxidize hydrogen sulfide and other reduced sulfur compounds to elemental sulfur, while some other members of thiobacillus which are highly acid tolerant, which can live in the presence of high concentrations of acid, they form sulfuric acid. They produce sulfuric acid from the oxidation of elemental sulfur or other sulfur compounds, and this generally occur under aerobic conditions. Thiobacillus denitrificans, it carry out sulfur oxidation under anaerobic conditions by utilizing nitrate as the terminal electron acceptor. So it can carry out sulfur oxidation under anaerobic conditions in the absence of oxygen. Hydrogen sulfide is also phototrophically oxidized under anaerobic conditions by photosynthetic sulfur bacteria. Bacteria belonging to the group Chromaceae, Chlorobiaceae, Ecothiorodospiraceae, etc. are examples for such photosynthetic sulfur bacteria. Among these organisms, Chromaceae store sulfur globules intracellularly, while Chlorobiaceae and Ecothiorodospiraceae they excrete sulfur globules. Some cyanobacteria also participate in the phototropic oxidation of hydrogen sulfide. So we were talking about oxidative sulfur transformation. Now, reductive sulfur transformations. Examples for reductive sulfur transformations by microorganisms are sulfite. It can be reduced to sulfide by a wide variety of microorganisms. For example, Ultramonas, Clostridium, D-sulfovibrio, D-sulfotomaculum, etc. A bacteria known as D-sulfuromonas acetoxidans. It can grow using acetate under anaerobic conditions by reducing sulfur to hydrogen sulfide. Some extremely thermophilic anaerobic archaebacteria such as Thermoproteus, Pyrobaculum, Pyrodictium, etc. are capable of sulfur respiration by using hydrogen gas. Some species of Bacillus, Pseudomonas, Saccharomyces, etc. are also capable of releasing hydrogen sulfide from sulfate. What is dissimilatory sulfate reduction? Some obligately anaerobic bacteria carry out a process of sulfate reduction known as dissimilatory sulfate reduction, and they are also known as sulfate reducers or sulfidogens. Examples of such sulfate reducing bacteria are D-sulfovibrio, D-sulfotomaculum, etc. They reduce sulfate to form hydrogen sulfide. The production of hydrogen sulfide have an adverse effect on living population in any habitat. Hydrogen sulfide is highly toxic to aerobic organisms since it could react with heavy metal groups in cytochromes. As a result, hydrogen sulfide kills nematodes and other microbial populations in waterlogged soils, and it could destroy plant roots and can kill plants. H2S also have a significant antimicrobial activity. So in a sulfur cycle, various forms of sulfur are cycled among themselves. Reduced forms of sulfur, oxidized forms of sulfur, and elemental sulfur are cycled through these different forms through different microbial actions. So there will be reduction reactions, oxidation reactions, decomposition and desulfurization, assimilation of sulfur, sulfur disproportionation, etc. All these reactions are carried out during a sulfur cycle. Now we are going to discuss phosphorus cycle. Phosphorus cycle is a biogeochemical cycle that describes the movement of phosphorus through various spheres such as lithosphere, hydrosphere and biosphere. The phosphorus cycle is a sedimentary biogeochemical cycle. There is no gaseous forms involved. The phosphorus reservoir is mainly in the form of rock deposits. There are no gaseous forms in the phosphorus reservoir in the biosphere. Phosphorus is an essential component of cell in DNA, RNA, ATP, phospholipids, etc. In environment, phosphorus is present as phosphates of calcium and iron, and generally these are not soluble, they are generally insoluble. This image represents a phosphorus cycle in which there will be mineralization, immobilization and solubilization of different phosphate forms. In soil, the source of phosphorus is the dead remains of plants, animals and microorganisms. Phosphate can serve as terminal electron acceptor in the absence of sulfate, nitrate or oxygen. Microbial transformations of phosphorus is done from simple orthophosphates to more complex forms of polyphosphates. Microorganisms can reduce phosphate, and the final product of phosphate reduction is phosphine. Phosphine is a volatile and toxic gas, and it ignites in the presence of oxygen and produces a green glow. Phosphine is generally liberated from swamps, soils and marine regions where there is extensive microbial decomposition. The phosphine ignites when exposed to air, and it can also ignite methane and give rise to a ghostly light phenomenon. The phosphate which enter the aquatic ecosystem becomes a part of the marine sediment, and it will be present there. It will get deposited there. So there are different steps in a phosphorus cycle. All are operated by soil microbial population. Mineralization is the conversion of organic phosphorus into insoluble inorganic phosphates. Solubilization is the conversion of insoluble inorganic phosphates into soluble inorganic phosphates. And microbial activities may also immobilize phosphorus. These are the different steps involved in a phosphorus cycle. So what is phosphate mineralization? The mineralization of phosphate occurs with the help of the enzyme phosphatases and phytases. Phosphorus mineralization is carried out by microbial population. Many soil microorganisms produce enzymes that attack the organic phosphorus compounds in the soil, and they release inorganic phosphate. The organic phosphorus are present in the organic matter from dead remains of plants and animals, and also from animal excretions such as urine and fecal matter. Some bacteria and fungi are capable of producing the enzyme phytase, which releases soluble inorganic phosphates from inositol hexaphosphate or phytic acid. Coming to phosphate solubilization, Bacillus, Micrococcus, Pseudomonas, etc., fungi such as Aspergillus and Penicillium, and mycorrhiza, they are involved in the solubilization of phosphate. The insoluble or inorganic phosphate is converted to soluble inorganic phosphate by this phosphate solubilizing microbial population. So the presence of phosphate solubilizing microbial population increases the availability of assimilable phosphate for plants. The mechanism of phosphate solubilization is mainly by the production of organic acids. Nitrosomonas and thiobacillus, they are capable of producing nitrous acid and sulfuric acid respectively, and they solubilize inorganic phosphate. The phosphorus solubilization by microorganisms is affected by a number of factors such as temperature, pH, aeration, carbon nitrogen sources, etc. In marine environments, the availability of phosphorus depends on the temperature of the surface water. Microbial activities may also immobilize phosphorus. The assimilation or incorporation of phosphorus into cell constituents will make the phosphorus biologically unavailable. The phosphorus assimilation into cell constituents remove phosphate from the bioavailable pool. In ocean also, when the phosphorus containing compounds in marine organisms, when they sink to the ocean floor, they will form sedimentary layers there, and as a result, the phosphorus present in the sediments in these marine organisms, the dead remains of these marine organisms, they also get not biologically available. Over long periods of time, these sedimentary rocks may be moved from the ocean to the land by a process known as uplift, but this is a very slow process. The phosphate concentration in an ecosystem highly influences the primary productivity. Addition of phosphates from detergents into lakes result in eutrophication. This will make the lakes eutrophic. There will be algal and cyanobacterial blooms. As a result, there will be an increase in the organic matter in the water body, and when this organic matter gets decomposed, the decomposition of organic matter will deplete the oxygen from the water body, which is detrimental for the living organisms present in the water. It may result in fish kills. So in a phosphorus cycle, different forms of phosphorus such as organic phosphorus, inorganic phosphorus, soluble phosphorus or insoluble phosphorus forms are all cycled in between by various microbial actions, which includes mineralization, immobilization, solubilization, etc. So in this session we discussed about sulfur cycle and phosphorus cycle. Thank you so much for listening.